I'm going to talk about targeted protein degradation for AML today. And I'm particularly interested in this because I'm a clinician who deals almost exclusively with AML. And we've had a few noteworthy new drugs approved for AML in the last couple of years. But prior to that, when I was training, we'd really been using the same agents to treat AML that we've been using since before I was born. Which was in stark contrast to the success stories of targeted protein degradation to treat myeloma. Eileen's already been over this. Just to reiterate the point, thalidomide analogs recruit new substrates to the CRBN complex. And it was in 2014 that Ben's lab and Bill Kalin's lab described IKSF1 and 3 as being critical targets for degradation. The following year, Ben's group described casein kinase 1 alpha as being degraded by lenalidomide. And of note, in 2016, CC885 was described as the first thalidomide analog that was able to degrade the other described substrates, but in addition, able to degrade GSPT1. Now, in their paper, they showed efficacy against multiple cell lines from multiple tissue types. But they've really pursued this as a treatment for AML. And as Ben alluded to in the introductions, there is some early phase clinical trials of the clinical grade compound CC90009, which have shown, I think, some encouraging efficacy in these studies. At the time, though, apart from degrading GSPT1, there wasn't much known about what the effects on the cell would be, which molecular subtypes would be best targeted in AML, and that was the basis for the start of our studies. So GSPT1 is often just described as a translation termination factor. It certainly is that, but it has multiple other roles. And so we started by doing a tiled CRISPR screen to see if we could identify the critical domains of GSPT1 that, when lost, lead to cell death. And we started this based on a paper by Shi et al. And basically the premise of the screen is that when a guide and Cas9 cut, about a third of the time you'll get in-frame repair. Now, if that's, you'll lose a couple of amino acids, but it remains in frame and you get the rest of the protein. Now, if that happens in a non-critical domain, then it behaves as a heterozygote. If it happens in a critical domain, then it looks like homozygous loss, and you get disproportionate dropout of CRISPRs, which are targeting that area. So it's just a brief overview of our screen. We designed a guide library which tiled every PAM sequence in the coding region of GSPT1, and then sampled cells at different time points, put them through next generation sequencing, and then looked at which guides were dropping out of the screen, suggesting they'd hit a critical region. So this is an overview of what GSPT1 looks like with the functional domains. And when David Chen, who was helping with this screen, he operates out of the City of Hope, he looked at the gray area here, the GC rich area, and said that's off target effect straight away. The area of GSPT1 that's been implicated as being binding to tumor suppressor genes, genes involving apoptosis and nonsense mediated decay, didn't drop out at all. And then when you go to the C terminus, it's the GTPase domains and the ETF1 binding domain that seem to be critical. And this fits with GSPT1's role in translation termination, where ERF1 or ETF1 mimics a stop codon, mimics a tRNA, I should say, recognizes the stop codon, and then GSPT1 lends its GTPase activity to terminate the protein. We wanted to validate this, and so we generated some GSPT1 overexpression vectors, including some isoforms. GSPT2 looks exactly the same, essentially, apart from not having the GC-rich area, and then a short isoform, which really only has the GTPase domain and the ETF1 binding domain. And we also generated drug-resistant constructs. Celgene had reported that G575 was a critical binding point, and the dogma is from the zinc finger structures as well as GSPT1 that this critical glycine is absolutely required. We did try and generate drug resistant mutants changing G575 to an alanine. Actually, that does degrade, and I don't have the data here to show you because it's a bit of a side step, but it's quite interesting that actually CC885 appears to be able to degrade with other amino acids in that position. However, for this story, it was all of the different isoforms we expressed were able to fully rescue, backing up the GTPase and ETF1 binding as being critical. We then interrupted this binding. We went to yeast biology and looked at the ETF1 binding surface, and were able to pick out 5 highly conserved amino acids, which when we disrupted meant that GSPT1 was no longer able to pull down ETF1. And these mutants, these GSPT1 mutants, were no longer able to rescue the phenotype. So this backed up our idea about failed translation termination. The question is, do you actually get failure of translation termination? And so we built these constructs where BFP has a stop codon through a linker into GFP. And so if the stop codon is not recognized, you get GFP expression. We validated this using an aminoglycoside where we saw dose and dependent increase in GFP expression, as well as the expected hierarchy of stop codon recognition where TAA is actually very difficult to read through. CC885 is able to read through TAA to some extent, and we get a dose and time-dependent increase in expression of GFP, implicating failure of translation termination. Oh, sorry, this is, I need to put my password here, otherwise it won't let me carry on. Okay. What's happening downstream of that? The labels have fallen off, but these red dots on RNA-seq are ATF3 and ATF4 and show activation of the ISR. I should point out that Celgene have actually produced a very nice blood paper where they've also demonstrated the critical importance of the activation of the stress response pathway. However, we were interested in this as a therapeutic angle, and if you're dealing with AML, nothing's quite as bad as TP53 mutated. And Stefan Betcher, who was in the lab at the same time as me, had a really nice science paper in which he generated some isogenic cell lines. And we used these, and we're able to show that the cornerstone of AML therapy, cytarabine and daunorubicin, have a very blunted response with TP53 mutations. CC885 didn't really seem to care about the P53 status. I'll come back to that at the end. No talk about thalidomide would be complete without mentioning the horror stories from the 1950s and 60s, and it's well described and very elegantly demonstrated by Emma Fink in the Ebert lab, the importance of I391V humanization for effective degradation of casein kinase with lenalidomide. And Celgene had shown that V380E was critical humanization for degradation of GSPT1 with CC885. However, we did a lot of other humanization. There are other areas you could humanize. We did all the combinations of those we could get. And one interesting thing that we found was that for casein kinase, for example, if you humanize I391V or V380, you do get some degradation, but really for maximum degradation, you need both humanizations. And further, if you go on to look at CC90009, the clinical grade degrader, you need both humanizations to get any degradation of GSPT1. And this is borne out in kill curves on BAF3 cells overexpressing different humanizations. And based on this, we thought that the minimal optimal design of a humanized mouse for investigating GSPT1 degraders was a V380E I391V double humanization mouse, which we went on to make. We generated MLL AF9 leukemias on this double knock-in background. CC885 is slightly tricky to deal with for in vivo experiments. So the first experiment we did was just a proof of principle, which was an in vitro in vivo study where we pre-treated for 4 hours with the drug, washed it off, and injected 10,000 viable cells, leukemia cells per mouse. And we were able to demonstrate a significantly decreased disease burden in peripheral blood and a survival advantage. Obviously, this isn't as good as doing an in vivo experiment, a pure in vivo experiment. So we've gone on and recapitulated much of this data with leukemia and CC90009, but of interest, we wanted to know what the effects on normal hematopoiesis was. So this is an experiment that Adam Sperling performed, and this is a bone marrow chimera between CD45 2 double knock-in and CD45-1 wild-type cells, followed by treatment with CC90009. And you can see that the peripheral blood chimerism is affected. We have some evidence that this actually does plateau out. But what was encouraging from our point of view was that if you look at the long-term HSC compartment, which would obviously be important for regeneration post, say, a cycle of chemotherapy, these were relatively spared compared to the other stem cell compartments. And we're continuing this work by doing some serotransplantations with these treated cells to show that we can adequately repopulate bone marrow in a transplanted mice. I suppose the question is, why should these be relatively protected? And we think that this is probably down to, again, to translation. So the stem cell compartment is, compared to a tumor, relatively low in translation. It's highly regulated, but relatively low. And we had done genome-wide, jump forward, genome-wide CRISPR screens and pulled out, obviously, cereblon. We pulled out some negative regulators of the mTOR complexes, but we also pulled out EF4A1, which is a key translation initiation factor. And actually, this was one of the most robustly validated hits in our screen. And again, the idea being that in the face of a defective translation termination, having less translation protects the cell. And we were also able to demonstrate a modest effect when we co-treated with amacitaxin, which is a drug which limits translation elongation. Coming back to the p53 story in our mice, we've crossed these mice, our double knock-in mice, with TP53 null mice. And whilst we get the expected blunted response with nutlin, we can demonstrate effective killing in the TP53 mice on treatment with both CC885 and CC90009. So just to conclude that, the bulk of the talk, which is that GSPT1 degraders are a new therapy for AML, I hope. Trials ongoing. We've demonstrated some new mechanisms of action and resistance, which may lead them to be effective in the highest risk disease. And possibly related to translation, long-term HSCs are relatively spared. And we've demonstrated some new models in which to test GSPT1 degraders in defined clinical or mutation context. However, this is all a little bit unsatisfactory, you might think, because we target GSPT1 because we can. I don't think anyone ever really went out to try and degrade that. In fact, when I was starting this project, Nico Toma was visiting the lab and he said, "Why are you working on that? It's pretty much the last thing anyone would want to degrade." I think his approach has softened somewhat since then. But it's right, we're doing it by accident rather than design, and just very, very provisional data that we've generated in the lab in the last 2 weeks, because we've only just recently opened after lockdown. PROTACs don't need any introduction, but they are a way of targeting. There's a whole number of PROTACs being described for targets, many of which are important in AML. But we wanted to target different things, and we were looking at gain of function mutations. The trouble with that is, of course, is finding something that only targets the gain of function mutation might be quite tricky. But we sort of reasoned that it might also be the leukemogenesis might not just be the gain of function, it might be the haploinsufficiency. And so even if you lost the wild type, losing the other copy might be deleterious to a cell. And there's some rationale for that from the studies that Ben's group did in casein kinase, whereas a haploinsufficient cell has a growth advantage. But if you degrade the other copy of that, or mutate the other copy of that, the cells actually die and have a proliferative disadvantage. And so taking that forward, because this data is so provisional, and I'm not sure how close this meeting is, I'm afraid I'm not going to tell you the target, and I'm not going to give you the structure of the drug. We've generated some PROTACs against gain-of-function mutations. Now, these are the first iteration of the molecules, so the degradation is modest. I accept that. Although this is a vastly overexpression system, it's difficult to know how the drug would cope with endogenous levels. But regardless of that, we are able to demonstrate a rapid response to these drugs with differentiation markers appearing, morphological evidence of differentiation, and a rapid expansion of cell numbers followed by cell death. So we're hopeful that some of these molecules might bear out in further studies. So just briefly, we have some new candidate PROTACs to target gain-of-function mutations, which do have desirable biological effects. I think I'll leave it there. There's a whole list of people to thank for this work. You know who you are because you're on the paper that's currently in revision. But I should highlight, obviously, Ben and Adam and Mikolai, who have been absolutely instrumental in driving this project forward, particularly in my absence overseas. And with that, I'll finish. Thank you.